Welcome everyone, and thank you for joining us for this short session. We are here to talk about hand hygiene. You probably already know why hand hygiene is important. We collect microbes on our hands by coughing and sneezing into them, touching garbage, handling luggage, and touching frequently used surfaces. Then, with the same hands, we touch our eyes, mouth, and nose. This provides a great opportunity for bacteria to find a way to infect us. There are thousands, even millions, of tiny creatures on our hands. Most of them are bacteria, but viruses and fungi can also be found on our skin. Today we will focus on bacteria. When I was visiting a cruise ship last time, I tried to measure how many bacteria could be found on frequently touched surfaces. Do you have any idea how we can count bacteria on a surface? For example, if I wanted to know how many bacteria are on those tongs, how can I measure it? The problem is that bacteria are so small that we cannot see them with the naked eye. So, the easiest way is to use a so-called contact plate. It contains a jelly-like material made from agar, the same agar that some of you might use in the kitchen. In that jelly there is everything a bacterium needs: water, sugar, and amino acids. When I touch a surface with the plate, bacteria from the surface will stick to the jelly. And once a bacterium is there, it finds that it has everything it needs. Do you know what bacteria do when they have everything they need? They start to multiply. So, one bacterium becomes 2, then 4, then 8, then 16, and so on, until they form a huge colony, so large that we can see it with the naked eye. It's similar to how cities grow over time, but in the case of bacteria, the entire process takes only 1 or 2 days. That's what I did, and I found this. Keep in mind that each so-called colony, originally came from one bacterium. If we try to count how many colonies are in the picture, we can estimate how many bacteria were on that surface, almost in your food. As you can see, plenty. I sampled other surfaces, like door handles. For that, I used a contact slide, which contains the same jelly material, just in a different shape. I also tested elevator panels and door handles in public restrooms. As you can see, I found plenty of bacteria everywhere. What do you think? Are they dangerous? That's a tough question. It's like asking if the fish in the sea are dangerous. The answer is similar. Most of them aren't. They're fascinating, and some are even tasty, so we're glad they're there. It's the same with bacteria. There are good bacteria that actually protect us, and we're happy they live in our skin. We don't want to harm them. These are called the normal skin flora. However, there are also harmful bacteria, which we call pathogens. Pathogens are the ones that make you sick. Interestingly, with bacteria, there's a third category: opportunistic pathogens. These bacteria won't cause you any problems as long as your immune system is strong enough to keep them under control. But as soon as your immune system is weakened for any reason, they seize the opportunity to infect you. This means that you might be fine with a certain bacterium because you're young and healthy, but if you pass that same bacterium to someone who is elderly, has diabetes, or has a medical condition that weakens their immune system, it could make them sick. Of the many bacteria I showed you earlier, most were probably part of our normal skin flora, and they're generally harmless. But when I sampled this rice spoon, I noticed something interesting. Do you see that strange green aura around a colony in the upper picture? We sent it to the lab, and they confirmed it's Pseudomonas aeruginosa. Pseudomonas aeruginosa is responsible for numerous human infections, affecting various parts of the body such as the skin, ears, eyes, respiratory tract, and urinary tract. In Europe, Pseudomonas aeruginosa isolates show a high level of antimicrobial resistance, with approximately 30% resistant to at least one class of antibiotics. This major human pathogen is commonly associated with recreational waters. The image shows Pseudomonas aeruginosa levels in various recreational water sources in the Balearic Islands, one of the most popular tourist destinations in Spain. As you can see, Pseudomonas aeruginosa is widespread in the environment, not only in water systems, but also in soil and occasionally on our hands. These bacteria are all around us. So how can we protect ourselves and prevent them from causing infection? The answer is hand hygiene. There are 2 types of hand hygiene. One is washing our hands with soap and water, which physically removes germs from our skin. The second option is rubbing our hands with an alcohol-based hand rub, which leaves the germs on your hands, but they will be dead. Both methods are effective. Which do you think takes less time to perform? Rubbing your hands takes about 30 seconds, while proper hand washing with soap and water takes around a minute and a half. So, rubbing is 3 times quicker. Which do you think is more effective? This is a tricky question. In general, rubbing your hands with sanitizer usually gets rid of germs more effectively than washing with soap and water. However, some germs are less sensitive to alcohol. For example, Clostridium spores are resistant to alcohol, and norovirus is also less sensitive to it. In these cases, physical removal through washing is more effective. As norovirus is especially common on cruise ships, washing hands with soap and water is recommended after using the restroom and before eating to help prevent infection. Also, if your hands are visibly soiled, alcohol may not be able to penetrate the layer of soil, so any visible dirt must be washed off with soap and water. But in general, rubbing is more effective. Which one is more skin-friendly? It may seem surprising, but studies show that alcohol-based sanitizers, when used properly, are more skin-friendly. But be cautious. Never apply sanitizer to wet hands, as that can cause skin problems. Your hands should be completely dry before using an alcohol-based product. How does hand hygiene work? Remember that we talked about good bacteria, and we mentioned that we don't want to harm them? Luckily, these good bacteria live in the deeper layers of our skin. When we touch something and pick up bacteria, those bacteria will only be on the top layer of our skin. We can easily wash them off or kill them with alcohol. Meanwhile, the good bacteria are safely hiding in the deeper layers of the skin, so they remain unharmed. This means that we can perform hand hygiene as many times a day as we want, without harming our healthy skin flora. I'd like to show you an experiment. We used the same contact plate that I showed you earlier. I touched the left side of the plate with my unwashed hand. Then I rubbed my hands with sanitizer and touched the right side of the plate with my sanitized hand. Can you guess what we'll see on the plate after a day? On the left side, we see how many bacteria I can spread by a touch without hand hygiene. On the right side, we see how many bacteria I spread after sanitizing my hands. The difference shows how effective hand rubbing can be. Here's another experiment. This plate contains a bacterium called E. coli. What I'm doing here is touching the E. coli plate and then touching 6 other plates that only contain sterile agar. Can you guess how many of those 6 plates will be contaminated by E. coli? All of them. See how easy it is to transfer bacteria with just a single touch. And what do you think will happen if we repeat the experiment with 50 plates? Yes, the same result. All 50 plates will be contaminated. Now, imagine someone on this ship is sick and sneezes into their hand, or doesn't wash their hands after using the restroom. Then they start walking around, touching railings everywhere. Remember these images, even after 50 touches, they can still spread germs. This shows how one sick crew member or passenger can potentially contaminate the entire ship. That's why hand hygiene is so important. Thank you for your attention.